Hi, I'm Anthony Murphy. I'm the CEO of Causeway Sensors, and we are developing novel nanophotonic sensors that will enable the future of pharmaceutical manufacturing. Causeway Sensors are a spin-out from Queen's University Belfast with a stellar track record of academic research in the fields of nanotechnology and plasmonics. And when we were studying the intersection of physics and biology, the founders seen the need for better sensors in the field of biomanufacturing. This opportunity has helped us raise 3.3 million in venture funding, and we've recently closed the seed round, which gives us an 18-month runway. With this money, we've assembled a high-class team with PhD level scientists and executive level experience in corporate finance. And additionally to that, we have a really strong board of directors with a history of exiting companies in our space, and we can lean on them for strategic guidance. We're operating in the pharmaceutical industry worth over $1 trillion in 2020. The market for biotherapeutic drugs is growing with over 10,000 new drugs in development every year. The fastest growing sectors are cell and gene therapies, closely followed by monoclonal antibodies. Causeway Sensors would compete in the subsector of process analytical testing, that is, instruments for controlling and analyzing bioprocessing, which includes pH, temperature, and mass spec. It's a massive market worth $3 billion, rapidly growing. Industry 4.0 is the latest industrial revolution, where the productivity of manufacturing can be greatly increased by the introduction of interconnectivity, embedded sensors, smart automation, and machine learning. The pharmaceutical industry, for a number of reasons, has been slow to move to Industry 4.0 standards. One of the most limiting factors is the lack of integrated sensors within a bioproduction facility. Schematic below shows the current process for obtaining affinity data. This data is crucial for determining the interaction of a new drug with its target. Drugs are grown in a bioreactor by genetically engineered cells. To analyze the affinity data, a sample is taken from the bioreactor, taken to a lab to be filtered, and then purified on a different machine, before finally making its way to the biosensor characterization lab where the data is generated. It can take 12 to 48 hours to get this data back to bioreactor operators, at which stage it can be too late to alter the process conditions. Therefore, the whole process is unoptimized and very slow to spot when things go wrong. Causeway Sensors are developing a next generation nanophotonic chip capable of detecting biological entities like antibodies in real time. The gold nanopillars resonantly absorb light at a certain frequency. When an antibody attaches to the surface, the resonance frequency is altered, and this is detected in the reflected signal. Causeway have engineered tailored optical, electronic, and microfluidic architecture that condenses lab-scale processes into a chip. The chip is fitted with fluid connectors and slotted into a compact instrument that sits at line with a bioreactor with automated sampling capabilities. Extremely small volumes of titer are extracted from the bioreactor, filtered in line, and characterized within minutes. Critical data like antibody concentration and affinity can be monitored throughout a multi-week fermentation cycle without disrupting the growth equilibrium inside the bioreactor. The Causeway Sensors nanophotonic chip and instrumentation can be used to monitor multiple bioreactors in parallel that feed back results to a central data hub. This real-time data, coupled with machine learning algorithms, will enable the optimization of biomanufacturing promised by Industry 4.0. Liberating key drug characterization data at the production line will enable better drugs to be developed faster and at lower costs. We have chosen MABS as our beachhead biotherapeutic market. It's a massive market already, with blockbuster drugs grossing billions of dollars treating cancer and autoimmune diseases. But more importantly for us, they need this affinity characterization data throughout drug discovery to large-scale manufacturing to ensure that the drug that they are developing is specifically binding to its target. Within the monoclonal antibody development cycle, we are further targeting the subsector of pilot production. At this stage of the process, you're trying to optimize the cell line and the process conditions for manufacturing the drugs at scale. We've gone out to the market and we've talked to customers to find out their pain points, and these are scalability, a great need to reliably scale from small volumes to high volumes, the ability to have this data online or at line, so currently this is done offline as discussed before. There's a lack of integration of systems, and the current process is really labour intensive. So the Causeway solution can address these, providing a fully integrated system that can monitor multiple bioreactors in parallel, that gives high value data quickly and throughout the pilot production cycle, and it's completely automated and touch free. We further detail the impact of our technology with this case study. In pilot production, several bioreactors run in parallel, and you often want to pick the best cell lines for scale and production. Due to constraints of the current biosensor technology, antibody characterization and affinity is usually only measured at the end of a 2-week fermentation cycle. With this data, it's easy to choose when a cell line is not performing, cell line 3 in this case, but it can be hard to choose between similar cell lines. Integrating the Causeway technology at line and giving more frequent affinity and concentration data means that better decisions can be made based on better data, and this can have a long-term impact when scaling production. We have analysed the potential market size of this beachhead opportunity. The TAM was calculated to be 70 million per annum by multiplying the number of pilot production programs running globally every year, being 3,100, by the amount the customer was willing to pay for this data at this stage of production. Similar analysis was done when expanding the market opportunity both downstream in commercial production and upstream in antibody discovery. And an even bigger market opportunity exists when expanded to different drug modalities like cell and gene therapies. We are exploiting this market opportunity with miniaturized integrated sensing technology. Our first prototype is pictured beside the Open SPR, one of the most compact devices on the market, and next iterations of our technology will see further miniaturization so that they can easily sit at line with the bioreactor. And we will have this prototype at our exhibition booth at SLAS Europe at the end of May. I also show some initial validation data coming off this prototype, where nanometer sized biological layers are easily detected by the system in real time. And here I present the product development and funding timeline for this product. As pictured on the previous slide, we are testing our first prototype internally. We will iterate and miniaturize this prototype further via this internal testing stage that's going on at the minute. We will then validate externally and target blue chip pharmaceutical companies and bioreactor companies to test this system out on, before full product release is expected at the end of 2023. Our most likely route to market will be via partnerships or OEM relationships with bioreactor manufacturers, and our latest seed funding round, along with a Strength in Places grant, means that we have enough funding and runway to fully validate this first product. We will need it to raise a Series A round to commercialize the product, and after successful validation of the first product and more investment, we can look to expand our products as discussed earlier with the TAM. So to quickly summarize, we are Causeway Sensors, a venture-backed company built on world-class academic research with a high-class team. We're developing novel integrated sensors that allow better data collection and bioprocessing. Our first product will target pilot production of monoclonal antibodies and be released late 2023. In the near future, we will be looking for a corporate-led investment to fund the company growth.